presentation. We'll move to the second presentation. And this is Professor Hari Krishna Koul, Professor, Department of Urology and Cancer Center, LSU, LCMC Cancer Center, USA. And he's going to talk on tumor progression and therapy resistance, DNMTs as potential therapeutic vulnerability. Welcome, sir.
Good morning, everybody. Thank you for the kind introduction. This morning I will walk you through two intertwined topics. One is tumor therapy resistance. We have been hearing, actually you just heard a nice introduction, prelude to how tumors become resistant to therapy, but it's not that simple. There are a lot more, many mechanisms. And then I will walk you a little bit about our work on DNA methyltransferase inhibitors and show you some work in prostate cancer primarily before I make my concluding remarks. So I have no conflicts. The work has been funded by NIH over the years. So in terms of, if you look why patients die from current therapies, why we don't have therapy for any cancer at this point, primarily because of the two things. Tumors keep on progressing. There's a dynamic disease. It is not where you start, it is where you end. And then there's a resistance to all the treatments develops over time. Second, we have answered many questions and we have got some answers, but then we have many questions which yet remain to be answered and many answers that we have not questioned. We have a drug, you treat to the patient, you say there's a therapy resistance, move on, there's another drug. As a result, at a global impact scale, 18 million people each year get diagnosed with different types of tumors and 10 million people die from the disease. This is despite our current state of affairs. So there are two reasons for that too. We'll go in the detail a little bit later. You will now argue that, well, we don't have treatment options in developing world, especially in India, that everybody does not get the treatments. They are available everywhere else, but these are data from United States. These are data from 2023. If you look at very carefully, about million men get diagnosed with cancer each year, and about 322,000 of these men die. That is despite all the treatments we have available for them. And in women, it is no different. 948,000 women get diagnosed, 287, like 1/3 of the patients die of the disease even with best treatments available. So clearly we have a long way to go to make any impact. So I will argue that these cancer deaths are attributed to therapeutic resistance because we have developed over the last 50 to 100 years multiple treatments for every type of cancer modality, whether there's a mutation, whether there's a pathway that is deregulated, and yet we have to figure out a way to make good use of all of them. So majority of the cancer deaths not only come from resistance, but also because tumor keeps on changing. It's a therapeutic, what we call tumor progression. So I would say there are unmet needs for many of the people who treat patients here in the management of the tumors. We don't know which of the patients have an indolent disease where they have a tumor that does not require any treatment. Probably patient will live with it rest of their life in nice quality. But then there's a lethal disease. We have no ability to distinguish in many cases which patients will die from the disease. We do not know the molecular determinants of lethal cancer completely. We don't understand them. And we have some targets that have yet to be developed. But there's also, in terms of the work, lack of good biomarkers, early detection, especially in developing world. Many of the patients, as we heard since yesterday, they come at a late stage disease and then there's at that time point we can do much about it. And tumor metastasis, once tumor goes from its origin to different organ, it takes a fairly short period of time for patients to die. They die very rapidly. So if we can, they're all intertwined. If we can have good markers, one, that's where the work is needed. Second, if we can do early detection work. And third, if we can prevent the metastasis. So we have worked in many of these areas. We have a bench to bedside research program, hypothesis-driven disease models, research tools, use clinical data, online data, and the lab resources. In general, if I summarize tumor progression, if normal tumor cells have deregulated growth, loss of cell cycle control, contact inhibition, but then the cells start moving from their origin, they relocalize, co-localize, and become resistant to the therapies. So conceptually, if you put these concepts together, normal cells become indolent or locally dividing mutated tumor cells. They do not have growth control. They proliferate. They have a loss of local tissue controls. They evade death, and they can live in hypoxia. This is kind of features of every local tumor at first point. But then lethal disease is when these cells move from their origin. They acquire ability to go to new targets. They have more clonogenic activity. They are resistant to therapies. They lose metastasis suppressors. They have extensive survival. Not only survive in hypoxia, they also proliferate and grow in those conditions. So what causes what are the tumor development issues? We have viral infections which cause oncogene activation, tumor suppressor loss, inactivation DNA mutations, which is well established, and we can't prevent any of that. Many of these, maybe we have vaccines against some viruses, but we will get indolent or local disease. Then the second part, which is yet where we have to make a progress, is how the tumors progress, how does microenvironment play a role, how epigenetics regulates this process, how genomic instability is effective. So our work has been in this area in the second part, and we think those will lead to many actionable targets that we have not been explored yet. In general, for every tumor, the path I told you is right, but in some cases, as in childhood malignancies, normal cells itself have a genomic mutation which give rise to a directly lethal disease. And in some cases, this process is very slow. Like in prostate cancer, you have 10 years, 12 years, 15 years. Patient can live normal life despite cancer, but then in some patients, this is very rapid. Pancreatic cancer, you diagnose it 3 years, 95% survival. But no matter what you talk, the path is same. Whether it is rapid or it is slow, that is up to you to decide, of the tumor to decide. So you can make it slow. There's a chemoprevention, what you call, is slowing down this path. It is not completely blotting cancer, it is just preventing the progression. So in terms of prostate cancer, we heard yesterday, prostate cancer is not a lethal disease, you are fine. But in the United States, 288,000 patients get diagnosed each year with prostate cancer, and 34,000 patients die of the disease, which is still, and it is the second killer in the United States. One would argue people live there longer, and maybe that is the issue. Maybe we shouldn't live longer, and that would be another reason. But this is a lethal disease, and burden is there. And women, similarly, if breast cancer was different, it is the same ratio. 43,000 women die of 297,000. So there's a lot of indolent disease that is being picked up, yet there is a lot of lethal disease that gets missed up early on, even in those conditions, and patients die. So at molecular or a cellular level, normal glands, the cells start proliferating. Cells start proliferating. As long as the green area you see, the tumor is contained with the tissue, you can operate on these patients, debulk, use radiation therapy or any therapy you can. Purely, you're not really curing the basis of cancer, but you're removing the burden of cancer. But once the tumor develops advanced stage, it moves on, and there is very poor prognosis, no matter which treatment option you give to these patients. A case in point is prostate cancer. You have a Gleason grade here. I don't have a pointer ability to show you any pointer, but so as the prostate cancer progresses, there is this cellular deregulation takes place. But when you treat them with enzalutamide, which is anti-androgen of the highest order, you start developing this resistant cancer. First, it is still adenocarcinoma, but then you get this neuroendocrine phenotype and double negative or triple negative prostate cancer now, for which there is no cure. So which part is treatment induced or which part is general because of the disease is still being debated. But yet, we would know before advent of enzalutamide, we had about 3 to 5% neuroendocrine tumors. Now there are 30%. So clearly you are killing good guys, and it gives rise to bad guys. So now I have been tasked to overview these mechanisms of drug resistance for this meeting. One is increased drug efflux. You may have given drug to the patients, but cells are not taking it, it gets out. There are drug target shifts. There is modifying drug metabolism, deregulation of cell death pathways, activation of survival pathways, and the last part, cellular plasticity. And there is, in all these, we can exploit epigenetic alterations. So if you put all these pathways together, I can a little bit argue here with you how, or show you a way how epigenetics may be able to help us overcome many of these issues. So let's take each of them together one by one. What is drug efflux? There's a multi-drug resistant gene which gets turned on in the cells. Many of them are characterized as ABC cassette of transporters, and the ABC family has P-glycoprotein, MDR1, MCRP1, BCR, AGP2. So this is a class of proteins which gets expressed on the cell membrane, and then any drug you put on the cell, it pumps it out. It's a drug pump. It is not really complex. Our kidney cells already have it. Many liver cells have these pumps. That's how they survive. So it's a survival mechanism which is already present in many of the human cells being exploited by the tumors. Second is drug target shifts. An example would be topoisomerase 2 gene alterations. So now when the gene gets altered, these cancers become resistant to topoisomerase 2 inhibitors. Then there's a fluorouracil and thymidine synthase inhibitors. Again, they result in transcriptional inhibition of thymidine synthase. If there is no target, your drug will not work. Third example would be androgen receptor insensitivity. Cells start making AR, which does not have target binding site. So you can have splice variants. That is one mechanism. But the last mechanism is resistance to paxilites and other taxanes. You have mutations in beta-tubulin, which is the target. So if the target shifts, no matter how much amount of drug you give, patient is not going to respond. The second part would be your modifying drug metabolism. So all drugs get metabolized by two kinds of enzymes. One is phase 1 and phase 2 reactions. Phase 1 are these oxidation reduction hydrolysis reactions. Phase 2 will be your consumption and conversion reactions. But in general, CYP pathway, which is 450, glutathione S transferase, you heard a little bit, uridine conjugation, are all these enzymes cells use to detoxify itself. So many of our drugs that are used in chemotherapy for cancer require these pathways for activation. They are inactive till they get activated. And if you get alteration in many of the first phase pathways, you can lose drug sensitivity. But also if you get activated, these CYPs and this uridine diphosphoglycan, UGT and GST, they conjugate with your drugs and make them ineffective. So that's another mechanism cells use. Finally, there is cisplatin resistance, which is resistance in breast cancer because of mutations and modifications to the CYP genes. Survival pathways, you just heard a beautiful talk on apoptosis. And there are two ways cells die. One you call apoptosis, which is a programmed cell death. Another is autophagy, where cell starts eating its own organelles when it needs more energy. And finally, there are survival pathways, PI3K, mTOR. These have been well established. And because 70% of tumors have a PTEN loss. And so therefore, any activation of these pathways renders cell very live so they can keep on living. So if you look at all these pathways, you have seen drugs, compounds, stocks, a lifetime of last 30, 40 years, we have been hearing at least this drug works, it works in cells, then it fails in the patient. It works in patient 3 months after that it fails. Because all these are not static pathways, they are dynamic. Cell has to respond, like if somebody shoots a dart at you, you'll start running. Question is, can we stop you from running so you can be targeted? Now, putting this in the context of a cell, all these pathways don't work in vacuum, they work together, they are dynamic. You have PI3 kinase pathway, you have MDR, you have death receptors, you have apoptosis, survival pathways. All this interplay kind of helps cells, tumor cells, save themselves from our drugs. The second part would be cellular plasticity, which has been relatively a recent concept, as I told you. In response to drugs, cells change their shape. We used to call it EMT before, which is epithelial cells, because most of adenocarcinomas come from epithelia. They become more like mesenchyme-like or portable cells. But it is more than that, they're plastic. So cells keep on changing their shape back and forth. As long as the tumors are well differentiated from a pathologist point of view, you have a local disease, adenocarcinoma, therapy responsiveness, and excellent prognosis. And once you have metastasis, dedifferentiation, loss of targets, mesenchyme-like cells, and there's a poor prognosis. It's irrespective of any tumor type you take. Depending on where these pathways are much more advanced, those tumors have shorter lifespan, or those patients have shorter lifespan, versus where these are very slow growing. So if you can modulate these pathways, and I think I can argue that cellular plasticity can be targeted by these DN-methyltransferase inhibitors. So, and many other newer molecules, if we can limit, we don't have to do anything, we have to limit them from running away from their phase. So EMT is not our figure, it is borrowed figure, but there are multiple for students, multiple genes which are driver genes, twist, vimentin snail slug, ecadrenal loss is associated with from epithelial cells become mesenchyme-like and all that stuff. So putting this all together from cancer drug resistant mechanisms, if you look at all the periphery, you call cancer stem cells, cellular plasticity, and EMT, these are all plasticity mechanisms, aberrant activation of survival pathways, dysregulation of apoptotic, or your autophagy would be kind of survival mechanism. Then drug target shifts, increased efflux, and drug metabolism. These are the outcomes for where drugs interplay with the cell. But in between, in the bottom and inside of this yellow circle is these mutations at genomic level and the DNA repair, what you have seen, many of new drugs targeting DNA repair. But if you see this epigenetic modifications, without an epigenetic modification, none of these processes can take place, because that is critical for cells to change their gene expression. Without epigenetic modifications, gene expression cannot be changed. So why can't you target it? So why epigenetics? Other thing, gene regulation is very simple. This is our human body. This is from NIH Common Fund, images from NIH Common Fund. So our body has about 37 trillion cells. All of them have the same genome. Each cell in your body has the same genome. Yet, skin cell is nothing like liver cell, nothing like kidney cell. So what causes this thing? This is just epigenetics, how the genes are, which genes are turned on and which are turned off. So you take 200 different cell types are in the body. And yet, you can see, look at this, you are seeing here, somebody shows you cancer cell becoming like neuronal-like, it becoming epithelial-like, but they are already in the body, that's what body does. Cells become what they want to become, and how they become is by regulating gene expression. And epigenetics is one of this mechanism, very cute mechanism of regulating that process. And when you call epigenetic imprinting, that means a imprint is already generated on the DNA scale that this cell becomes skin-like cell. And you can also de-differentiate based on transcription factors to become more like stem cell again. So identical genetic information, yet different outcomes, is achieved by epigenetics. And therefore, I argue that epigenetic alterations in cancer therapy resistance may offer us most actionable targets. So there are now multiple ways to look at it, but within the short period of time, all I would tell you, our DNA is compacted on the right-hand side. When the DNA is compacted, gene expression is turned on. When DNA is opened up, which is on left side, we call it euchromatin, genes are turned on. So this process is regulated by group of chromatin modifying enzymes, DNA methyltransferases, ATP dependent, chromatin regulators, histone modifying, you can name them, and then chromatin condensation takes place with multiple other non-coding things. So all these different pathways are targetable. I will make a little point, another cartoon I will show you to make this simple. All this DNA, that's this double-stranded DNA, is packaged into cell, into these nucleosomes. You see these blue things. Thank you. And they get packaged into this, and methylation on the DNA or these histone tails regulates how things work. And so you can target any of these DNMTs. So since I have been given very few minutes, but I will try to make good use of it. And prostate cancer is not really a major problem for many people. It is BPH. When you get older, prostate becomes bigger, which is benign prostatic hyperplasia, so your urine outflow is blocked, and that's why patients usually go to a physician. But as long as the disease is within the organ, you can do any treatment, you can cure. Once disease has left, stage 3, stage 4, you have really no control on the disease, as I showed you. I think I would, because of the interest of time, I will help. So the long treatment time in prostate cancer, but if you look at this, after they become resistant, it is few weeks to months only survival. So all those treatments of $100,000, $200,000 on these patients is very few month treatments. While as this long disease, Doesn't matter what treatment you use, initially there's a good response to slow-growing disease. I will just skip through this because I don't think it will be justice to this or the project itself. So we work on a gene called SPDF, which is highly expressed in prostate, breast, and salivary gland, among other cells. It is a differentiating. Can you enter there, please? Excuse me? Can you enter? Just press OK. And we found this is a differentiating gene. This causes prostate luminal differentiation. Okay. This is all, most of the work is published, so you can read through this. Overall, what we have to say is SPDF is lost in advanced prostate cancer, and it has an anti-metastatic role. PDF is involved in cellular plasticity, and generally how PDF fits in this process of tumor progression. We have worked that pathway. Eventually, we have figured out, I was a young guy when I started the work. Now this is still not finished, but there's one gene that in entire prostate cancer we found that is differentially regulated between luminal and other variants of tumor. Let's skip through this and just get to the chase. So in clinical patients, as the disease progresses, you can see two different data sets. This PDF gene expression is decreased as the tumor is progressing. It is also at the protein level decreased, but this resistance is associated with, again, with the tumor therapy resistance. The neuroendocrine tumors have no PDF gene expression, and in cell lines, there's no difference. Aggressive cell lines have no gene. But if you take each patient and see their PDF levels, you can categorize them just by this one gene into either a neuroendocrine phenotype or the luminal phenotype. And at a gene expression level, you can see patients with low PDF have a neuroendocrine signature or the mesenchyme-like signature. To make this story short, you see there's a plasticity of the cells during that process. There is activation of mu pathways and loss of the differentiating pathways. Together with this, when we look at the clinical data, DNA methyltransferases are expression is increased, and this expression directly regulates the therapy resistance, at least you can see in neuroendocrine tumor world. So this is at a scale of the single gene. Now looking this further, diving deep into the gene expression data on the PDF promoter and enhancer region is methylated, and methylation on it is directly regulated to loss of its expression. In cell lines, you can see that there's a direct inverse correlation between DNMT expression and PDF RNA expression. I think it is. So using the DN methyltransferase inhibitors, you can reprogram the neuroendocrine cells. You can treat these patients for 15 days or treat the cells in vitro or the tumors in mice for 15 days with DN methyltransferase inhibitors, then treat them with the enzalutamide. You completely remove those tumors. And at a molecular level, you can see methylation on the DNA itself gets altered by using methyltransferase inhibitors. So we have ability to kind of modulate how cells respond to the treatments, not to change the chemicals that we are using or the agents we are using, but change cells' behavior, how they respond to them, and overcome therapeutic resistance. This, again, part of this is published, so I will not go there. So then we found that just using these methyltransfer inhibitors is as potent as using enzalutamide, which is the standard of care for these patients. So these are very low toxic, general, they're approved for other uses. Dextrostabine is being currently in the clinical trials for many tumors, but is approved for other conditions. So you can use these currently approved and less toxic drugs. Sorry. I think I will just stop here. So in case of, as the tumor progresses, there is more these modifying enzymes for the DNA and methyltransferation. You change the gene expression, and if you can block that, you don't kill the cells, but you block their ability to respond to treatments. And I thank you for indulging me and giving me a little more time. But epigenetics, again, I would argue for you, look at it at a more scalar level. Genetics is only the tip of iceberg. it is the epigenetics that really does the workhorse job, and so you can use all this for your own good. I thank my many of the colleagues. Thanks so much.